watch me bring the fire, set the night light. Shoes on, get up in the morning, cup of milk, let's rock and roll. Kink out, kick the drum, rolling on like a rolling stone. Sing song when I'm walking home, jump up to the top of the brown. Ding dong, call me on my phone, nice tea and I'll get my ping pong. This is getting heavy, can't hear the bass, boom, I'm ready. Life is sweet as honey, yeah, this beat chain like money.
So here, now let's take a look at the evidence of actual chemical evolutions. So as we went over in the previous session, to go through this chemical evolution, you have to be able to start from inorganic compound, and for somehow these inorganic compounds should be able to form as an organic monomers, and from that organic monomers the polymer should be generated spontaneously, all of them. One such event that can take place spontaneously is the cell membrane. Cell membrane is in chemically the composition of cell membrane is individual chemical component of cell membrane is a specific type of a lipid called phospholipid, and this is the structure, the model of the phospholipid is once again I put an emphasis on this. You don't have to worry anything about this chemical things, but simply I am showing this chemical structure to make my point across. So this is the individual chemical structure, basic structure of such a component, the individual component of the cell membrane, and its name is phospholipid, right? And this is a lipid, and why this is called a phospho, as it contains some specific chemical element called the phosphate. Phosphate is a molecule over here. This is a phosphate having 4 oxygen. Phosphate is phosphorus specific element is surrounded with 4 oxygen, and as you can see, whenever you have this oxygen, and this part is going to be polar charged, right, because of the presence of an oxygen. On the other hand, this part is a typical hallmark characteristic of any lipid, and the reason why the lipids are non-polar is they don't have any oxygens, and there is no room for any charge or being polar, because it all is made up of carbon bonding, carbon hydrogen, and this is actually basic chemical backbone for like oils, like petroleum and crude oils. So that's why they don't get mixed with water. So this is kind of the mixed bag of, unlike other typical lipids, this phospholipid is a mixed bag. Having both polar and non-polar part together in one chemical. So this part is non-polar. On the other hand, this part is a polar head group. So in biology, instead of using real chemical formula, individual phospholipid molecule is usually described as like this. So the big seemingly big head area is we call as a phospholipid, the phosphor head group, polar head. And this tail area we call as a non-polar tail group. So this single molecule will have this area is a polar and this is a non-polar. If you have that, then you will have a very specific unique chemical property will be featured on this molecule. So this polar head group versus non-polar fatty acid chains. This is actually the area that makes up a fatty acid non-polar part. And the reason why the cell membrane can have this clumping of all these many different phospholipid molecules stuck together is they are aggregated because of this specific environment of water, aqueous. Which means the environment surrounded with the water. Imagine if you dump so many different number of individual phospholipid molecules in the water, what would happen? Because this top area hydrophilic head is water loving, and on the other hand the other half is a hydrophobic water fearing area. And this particular arrangement was done spontaneously because now by forming this particular shape here, these are the water layer, water shell, and those water shells is faced with what? The hydrophilic head groups. Why? Because they can deal with water, because they are hydrophilic. And at the same time, the internal area which are occupied by hydrophobic, the fatty acid tail, they stuck together because by doing so they can avoid the contacting with the waters. And this is the most, energetically speaking, the most stable configuration. So actually if you do this, you will have instead of having decrease in entropy, rather they will create some environment where the increased entropy is observed because of this stability. If you somehow make the environment, the condition increase the entropy, then you are stabilizing that environment. Take it this way. So this is how. Let's imagine that you dumped so many different individual phospholipid molecules and then they will arrange by themselves. You don't have to tell them what to do, or you don't have to provide any extra incentives in the form of energy. They will spontaneously have this. Then inside this is water, outside this is water. In this way, all these head groups are the one facing and making contact with the water, and while this hydrophobic areas over here can enjoy their safe haven here, not making any contact with the water. So this is the most stable structure and energetically speaking, and that's why you will also have this increased entropy rather than every individual component of the phospholipids are being scattered around in the water. That's more actually unstable. So in other words, having this barrier in the form of cell membrane is no problem actually under the specific condition, the environmental condition. The only thing matters is whether this event of a spontaneous formation of a cell membrane also accompanied with the inclusion of this particular special molecule. We call it a self-replicating molecule. This self-replicating molecule, if some lucky membrane sac somehow get to include this self-replicating molecule as their thing, then now we are talking about this establishment of the genetics, the heredity, and what this complementary chain means. We are going to take a look at it, get to that in a minute. So that's the end of this chemical evolution. Once you successfully reach to this point, then you succeeded in creating a new cell, even if it is a kind of an incomplete pre-cell, but that's also as good as a cell. Then you can start a new horizon from then on. So that's the end of the story for the chemical evolution. Whether you believe it or not, of course, no one, even those even for one of the most strong proponent of this chemical evolution theory, they have never been there, so they cannot really for sure with 100% confidence that was the way the first type of living molecule, the cell, has been made. But this is some how reasonably all those scientists involved believe to have happened. Now, this chemical evolution theory, that's why, because the ancestral form of the first ever type of a cell, from then on, all these descendants were made through this activity of cell division. That's why this unity aspect we can observe in all different living organisms nowadays. So as we went through each different steps level of the synthetic step, from more from simpler molecule to a more complex molecule, each step actually seemingly involves the decrease in entropy. So it is viewed as an energetically unfavorable, so it must be a non-spontaneous reaction. So doesn't it violate the second law of thermodynamics? However, no, not necessarily. Under specific conditions, as long as under such condition energy can be provided spontaneously, then it can. So for that matter, actually, the particular environment of the early Earth, which we believed as the reducing atmosphere, might have a big role in such event to happen. Reducing atmosphere is something that the atmosphere that is something that not familiar to us as in these days. So in these days, all this our air contain plenty of oxygen, so oxygen containing atmosphere, but all the scientific evidence point that the early environmental condition of the Earth, they didn't contain any oxygen. No oxygen was present in the beginning. So that type of atmosphere we call as a reducing atmosphere. Because why it is called the reducing? Because the chemical activity involving the oxygen. If one chemical A get to combine with oxygen to become like AO, then we call that as an oxidized. Right. The opposite way of a chemical reaction, if somehow this element got away from the association with oxygen and became free like that, then this chemical we call it as a reduced. So with this aspect, we call that as an environment lacking the oxygen. We call it as a reducing atmosphere. And under such condition, probably the interference by oxygen is non-existent. So oxygen will try to break everything, because by its nature of the electron greedy and strong desire to have this taking electrons, it actually degrade and it breaks everything. But if you don't have any oxygen, that kind of destructive activity might have not happened, or at least at the least it would have been much less, so that it would have been much easier to have some specific synthetic chemical reaction in nature, is what the scientists believe. So this reducing atmosphere, a little bit more favorable condition to have this anabolic reaction. Anabolic reaction means synthetic reactions. So many scientists actually believe that such place might have been in deep sea vent, where very specific condition of temperature and some specific physical environment might have existed. So in the deep sea vent, or some other scientists believe the origin came from the extraterrestrial, so outer space. But in this case, actually, we are delaying the problem. So we will have to explain then what was the beginning in that outer space. So I'm not going to go into that discussion in this class. So this is the photograph of such deep sea vent existing even today. We call this the hydrothermal vent. And so look some. So with that, once you have established this, then starting from that, all those descendants were made through the biogenesis procedure. And as we have seen in the previous chapter, that's how we have this specific speciation, which we will get to there are a little more in the following part of this chapter. And this Darwinian evolution, as we have seen, is going to be responsible for all these different variation, like the diversity of organisms living today, because that's their nature. The gene, the DNA will change spontaneously over time, and any change in the gene will be manifested by the different traits. So here we get to see so many such diversity in the form of 3 different groups of living organisms. We call this 3 domains of life. They are domain eukarya, all the eukaryotic cells containing in organisms belong to this domain eukaryota. And as we have seen, the rather closer to this eukaryota, the one big prokaryotic group of organisms called the archaea. So these are all these archaebacteria. They are prokaryotes. And as you can see here, the branching point exists between the ancestral form of this archaea and the ancestral form of whatever they were, of eukaryotes. So that's what the scientists believe that they are as the origin of this evolution of eukaryotes. And a little far away from, more distantly related at this point, there exists this branching point between another group of prokaryote. We call this the present day of a modern bacteria. Okay, so many different groups over here. So we just simply ignore, not that they were unimportant, but it was too much complicated, so we don't have any time to focus on their the different diversity. But let's just take a look at ourselves, eukaryote, and these animals, obviously the one that probably most of us feel most comfortable and familiar with, yeah, including us. So here is one branching point from which three different groups of eukaryotes, like plants and animals and fungi. Fungi, as once again, includes the mushrooms and mold and yeast. They are all eukaryotes, right? But then what about the others? Whatever they are, ciliate, flagellate, who cares about? But all of the rest of these guys, what are they? They are the protists. These are the protists. So in other words, the group, protists as a group, is so big and diverse, actually. Although they may look relatively more primitive as compared with the other groups of eukaryotes. And another very interesting aspect of all these protists are, unlike the other eukaryotes, where we can easily pinpoint the branching point, the base from which different groups diverged. But it is very difficult to pinpoint any single common origin for all these different protists. There is no single starting point, which means we still have a difficulty in locating their actual origin among, is currently we are at the status. Okay, so like I said, all this diversity actually is the result of the mutation in the given gene, and the definition of mutation is any alteration, the change in the DNA sequence, or that change can also come from the larger scale alteration, like actual modification of a chromosome structure. So although this mutation is not really a very desirable thing, it's because the outcome, the end result of any mutation, generally speaking, is disastrous. The individual bearing any mutation will not be able to survive as healthy as the others who don't carry any mutation. However, still, the ultimate source of these variations are actually these mutations in the gene. And here, let's putting this Darwinian evolution aspect a little bit aside, and let's get back to this proof of the spontaneous generation in regard to the chemical evolutions, because no one was there, so it is very difficult. Although one can imagine that might have been the case, but how can you prove? Well, if one can simulate, or one can recreate such exact event under the similar in the simulation setting, then it might have been taken as a proof. So the first ever such experiment was done in early 20th centuries. In 1953, it's a very famous Miller-Urey experiment. And here the Miller, this himself as a graduate student pursuing the PhD degree, he independently somehow designed this experimental apparatus to demonstrate the spontaneous synthesis of organic compound, organic monomer, starting from simple inorganic molecule, is actually possible, and he did succeed. Especially, he was interested in whether that experiment can generate this spontaneous this amino acid, because amino acids are the building blocks of proteins, and obviously, as I have explained already, the importance of proteins, why it can be regarded as such an important component in building like matters. One such case is like enzymes, and so many other very important biological macromolecules are all provided by proteins. That's why. So, he built simulatory apparatus of that particular early Earth's reducing atmosphere, and here, so that in this big part of the space of a flask filled with only the primitive ancient Earth's atmosphere, like no oxygen but only having methane and ammonia and water vapors and hydrogen gases like that. And by he simulated the energy input by providing the electrodes, then imagining that an early atmosphere that might have some of the lighting might have been able to provide such energy source. And then he collected the some cooled down product at the bottom here, and then examined to see whether, so the starting material was all the gases, and here whether would he be able to collect some magic wonder component of organic monomers, was the purpose of this experiment. And then surprisingly, yes, he was able to. Also, disregarding all these things, the important thing is he was able to obtain this particular chemical component, which is the simplest amino acid. But no matter how simple it is, and it is the starting point, and it's very, very significant turning point of these whole issues. And later, by the repeats of this experiment by Miller himself, as well as with the others, other copycats experiment by other scientists, and actually all 20 standard amino acids. What I mean by the all 20 standard amino acid, although theoretically, like in nature, many different types of amino acid, perhaps several hundred different types of amino acid are possible, but the ones that participate in the making the proteins in the livings are only 20 kinds. And these particular 20 different types of all amino acids were actually able to be synthesized in this the laboratory settings like this. That means all different amino acids can be spontaneously synthesized in the early the primitive Earth. That means proteins can be generated spontaneously, not to mention that all these different carbohydrates. And we already know that the cell membrane can be spontaneously generated relatively easily, and with that matter, the nucleic acid is also possibly be generated. So why not, is the argument. And even better yet, actually, this sought after conditions of a so-called reducing atmosphere does not turn out to be not that critical. So even without having such a critical reducing atmosphere, the spontaneous generation of such important organic molecule was also possible in the laboratory. Was that? Yeah. And so this Stanley Miller was back then in 1953 was a mere graduate student under the supervision of this already renowned scientist Harold Urey, and both were recognized by this work. And so in continuation of this work in more complex organic polymers, and even those pre-cells were synthesized. Okay? So like this, the RNA absorbing the synthetic cell membrane like structure was also made at some time, and also some specific pre-cell inside of which even this simple metabolic process was observed as taking place spontaneously. So it seems it's almost there, this creation of new cells spontaneously under the laboratory setting, but that was it. Since then, no one was able to go further to actually show here is the actual more finished product of this spontaneously generated cell. No, it's not there. We don't know why. Still, so much more complex procedure has to be there, and also partly because of the lack of the incentives in the form of funding. All these scientists must be driven by the financially supported by the funding, and this type of works are not usually actually easily funded. So maybe that is the main reason. We don't know yet. On the other so that's what I'm saying. So no synthesis of an actual cell in a laboratory has ever been reported yet until now. However, that was actually the driving force of the new era of this so-called synthetic biology. So everything is in these days, some group of scientists will actively pursue this approach of creating artificially everything in combination with some other the invention of the engineering too. So that's the another very noticeable I shouldn't say noticeable, and also I shouldn't really say even promising, because that contains its own share of really the danger of practicing this synthetic biology. But anyway, it's there. So, for one such example of the example of synthetic biology, artificially, as I have mentioned in the previous chapter, that Craig Venter was the one who succeeded in generating totally artificial cells, starting from simply synthesized with the beginning of an artificially synthesized whole genome, like that. These are the result of the synthetic version of those cells. Okay, so here let's take a look at the property of the DNA as a genetic material. I have already mentioned that the special thing about the DNA is it features of the so-called self-replication capability, and that's why we take this DNA, the DNA, as take for granted as a genetic material. So let's take a look at what it means by the being able to self-replicate here. The DNA is named standing for deoxyribonucleic acid, and like I said, this is an organic macromolecules capable of self-replication. So this self-replication will make it ideal molecule for genetic material. And here comes the comparison between DNA and RNA. Both of them are the sole members of the nucleic acid, one major group of the biologically important macromolecule, organic macromolecules. The left side you get to see a model of DNA, where the right side is the model of RNA. So the difference between DNA and RNA appears to be one is a chubbier and the other one is a skinnier, right? The reason why the RNA appears to be skinnier while the DNA is fatter, chubbier, is because DNA is made up of two strands. Here they are described in two different colors. One is in gray and the other one is in yellow or ivory or whatever. Whereas the RNA usually prefer to be existing as a single-stranded molecule usually. So the nature of this particular nature of being able to exist in a double-strand form is actually key essence of capability of the self-replication. Anyway, let's take a look at the chemical component of this DNA first. As I said before, it is a long polymerase made of nucleotides, individual components of nucleotides. And the kinds of different nucleotides are only 4. So in this thread-like polymer, each component here, this nucleotide, and per each single nucleotide, there are 3 different parts. So together, combined together, of these 3 different parts now making one individual nucleotide is actually their way of organization in any nucleotide. So a single nucleotide consists of 3 parts. One is 5-carbon sugar and ribose here. So in 5-carbon sugar, there are 2 different kinds of 5-carbon sugar here, and here as I said, in the case of covalent bond, any solid line between the element is covalent bond. So here, this solid line means a covalent bond is established between this carbon and this hydrogen, just like that. And the 5-member ring-like structure, pentagon-like structure, at each point you don't see anything, but actually at each point carbon is omitted. That's the usual way of describing the organic molecules sometimes. So this one carbon is there, and there's another carbon is there, and another carbon is here, and carbon is there too. And also what you notice is at each position here, the carbon position is numbered, like 1' and 2', the carbon number 2, carbon number 3, 3', and carbon number 4, and finally carbon number 5. That's what we call this 5-carbon sugar. Its name is deoxyribose. This one particular simple sugar, monomeric sugar, like similar to glucose. In the case of glucose, it's a 6-carbon sugar, but in the case of this deoxyribose, this is 5-carbon sugar, carbon containing. But its close relative only appearing in RNA making nucleotide. So this deoxyribose, whatever the name is, only appear in the nucleotide included in the DNA. So only DNA making nucleotide contains this 5 carbon sugar deoxyribose, whereas only the RNA making nucleotide, the other type of sugar, ribose, appears. And if you compare the difference between deoxyribose and ribose, you will easily point out there is only one difference in this position. So like this, the component attached to this number 2 sugar, carbon number 2 sugar. In the case of a ribose, this is OH, which means oxygen containing, which we call this as a hydroxyl group. Anyway, whereas that oxygen is missing in deoxyribose, and that's why it is called deoxy. D means something missing. So that's the difference between ribose sugar, which is actually the prototype of this particular type of sugar, and the divergent group of this ribose sugar is in the deoxyribose sugar. And for some reason, in making DNA, only this deoxyribose sugar is used. But anyway, so getting back to the description of the component of any single nucleotide, yeah, this is the sugar present in every different nucleotide in DNA. Like I said, there are only 4 different types of nucleotide, and each 4 different types of nucleotide, the same old deoxyribose will be there. And the other part is made by another chemical we call the nitrogenous base. There are 5 different types of nitrogenous base can be possible, but in the case of a DNA nucleotide, only 1, 2, 3, 4. Oh, sorry. These 4 different types of nitrogenous base. One is called the adenine, and the other one is called guanine, and cytosine, and thymine. In the case of RNA, instead of a thymine, uracil is substituting the thymine. Actually, chemically speaking, no one has to memorize any chemistry, but like these are, so cytosine, thymine, and adenine, guanine, these 4 different type of nitrogenous bases are found in this part of DNA making nucleotide, whereas instead of a thymine, the other 4 different types of same nitrogenous base will appear on this part position of RNA making nucleotide. That's all. No further details is unnecessary for you to know. And last part is taken by this phosphate group. Phosphate we have already seen in the case of a phospholipid. So phosphorus surrounded by 4 oxygen, and this part, as you can see, the presence of oxygen having 2 negatively charged oxygen make each single nucleotide, regardless of actual kind, make it very extremely hydrophilic, negatively charged, strongly. Another point is that the uniqueness of each different nucleotide come from this position, nitrogenous base. Except for this nitrogenous base, the other 2 parts are all the same in 4 different types of nucleotide. That goes, same thing goes true to the case of RNA. So instead of naming the using the full the name of each nucleotide, we can usually simplify it in using the abbreviation of each nucleotide. So adenine nucleotide, we can simply put A to indicate adenine nucleotide, and guanine, instead of writing and guanine, but simply G and T, C, something like that. So this is actually what we mean by this DNA sequence in practice. In other words, when we refer to any DNA sequence, A, G, C, T, A, something like that, A, G, randomly, like these. Now you know what I mean by that. This is DNA sequence of in a particular area of the DNA. So this phosphate group provide DNA and RNA with a strong negative charge and make DNA and RNA highly polar and hydrophilic is this. Okay, I forgot about it. Another thing is, let's take a look at this ATP structure, revisited for a while. We have seen this structure of ATP previously, but now, what do you think? This ATP in structure, it has a ribose sugar, right? Because it contains oxygen containing hydroxyl group rather than deoxy. So this is ribose. So okay, one ribose sugar, and this is adenine nitrogen space, and you have this phosphate group. Although instead of one, you have three. So this is one legitimate nucleotide, although it is an RNA making nucleotide. Actually, when it can be incorporated into the RNA chain, perfectly okay it is. So this ATP actually has a dual function in that, and one, it is being utilized as a cellular energy currency, as an energy source by shedding off one phosphate to become ADP, being used as an energy. But at the same time, it can also be incorporated into the existing chain of RNA by shedding off two phosphate and to become one such component of an RNA. Yes. Now it's the highlight of this structure of the DNA in a relation with their feature of self-replication property is this: the complementary base pair. In this diagram, in figure, what you can notice is this model illustrates the way these two DNA strands are assembled. And what you can notice is, one thing is, in this structure of the so-called double strand of DNA, inside of this helix, double helical nature of the DNA strands, inside of the helix are occupied with all these nitrogenous bases, right? And also, more importantly, the relationship, pairing relationship among the different nitrogenous bases are very specific. So no matter where you look at, their pairing combination is always between A and T and between G and C, like that. And the dotted line over here, each dotted line represents the hydrogen bond. So, like I said briefly in the previous session, all these nitrogenous bases were held together through specific hydrogen bond, and the reason why always A, the nitrogen space A can only pair with a T, while the nitrogen space G can pair with only C, is because of their particular structural limitation or configuration, where like between guanine and cytosine, the three hydrogen bonds are possible, whereas between adenine and thymine, only two hydrogen bonds are possible. So it's just like you can think this particular relationship as like your electrical plug, like between Korea and the United States is different. So that in the United States, the two hole plug, whereas in Korea, three hole plugs electric. So that if you carry your electrical device to the other area and it won't fit and things, stuff like that. So that's why always this particular specific relationship do exist, and we call that as a complementary base pairing. So in complementary, why do you call this as a complementary? Because so having this relationship, now you know which is your partner. So like this behave as a mirror of, if this type of chemical interaction exists, then let's see, you have this AACGATCG, for example, in this particular DNA sequence. You know this is a DNA sequence because this T exists over here. If this was an RNA, instead of T, the uracil U must have been present over here. So, but then you immediately know, by knowing that DNA usually exists in pairs, double-stranded, then you immediately know the other part, or the sequence of the other guy, partner, should be TTGCTAGC. So by knowing only one part, you immediately know the other information is automatically provided, and in that way we call this as complementary. It has some kind of a mirror images like that. And why complementary base pair can serve as being capable of self-replication? Think of like you're trying to make up. In doing this, the activity of makeup, what would be one of the most important tool? Without this mirror, you cannot do anything. But if you have a mirror, then you can do this self makeup. Even on the subway, you can do this, right? So that's why it is called a complementary base pair. And in other words, so this complementary base pairs using this DNA exists as a double strand, and it enables this self-replication capability like this. So what I mean by the self-replication, it will be able to perform this replication without the intervention, without the help of specific enzyme. No, that's not what I meant. It requires the specific enzyme, in this case DNA polymerase. Is the name of an enzyme which required for this activity of DNA replication. However, each strand, all you gotta do is when it comes to this replicate, then you simply open up, temporarily disengage this relationship of a complementary base pairing, and then allowing this new synthesis of new strands is represented in green ribbon over here, and it can be easily copied because all you gotta do is follow the rule of this complementary base pair. So for each existing strand, represented in this sky blue color, all you gotta do is insert the corresponding complementary base pair carrying nucleotide. It is that straightforward. So each existing strand is serving as a template for the synthesis of new strand, is what it means by the self-replication capability. So once again, in summary, this complementary base pair of the relationship is the key for this self-replication capability of DNA double strand. And also, not only this, all the bright side of the fairy tales exist in complementary base pair, but also once scientists realized, actually it was first realized by the Watson and Crick when they came up with the correct structure of DNA double helix, and as a result they won the Nobel Prize. It can pass the spontaneous mutations onto the next generations. Why? Because each nucleotide can be converted into different one spontaneously, chemically it is. So like, for example, cytosine, you don't have to memorize anything, but cytosine easily can be converted into thymine through this chemical conversion process spontaneously in the cell. So if this event occurs just right before the DNA replication starts, then the newly synthesized complementary base pair will be inserted into the wrong, like originally the guanine should have been inserted in as a pair of cytosine during the DNA replication. However, now as this cytosine has been converted into thymine, then ADE will be inserted into the newly synthesized chain of the DNA synthesis. Then that is a mutation. So even though cells have their own different mechanisms of reducing and then checking some of the mistake happens during the DNA replication, still this type of spontaneous conversion can go undetected, so mutation will occur no matter what during DNA replication. And those alterations can be highly elevated. The rate of mutation can elevated by many different chemicals, and all those chemicals, group of chemicals that can cause DNA alteration, the nucleotide alteration, we call that as a carcinogens. Carcinogens means cancer-causing chemicals. So that means all those cancers are actually the outcome of the mutation, is what we can learn from this. So this mutation, we go further, this mutation things, and during DNA replication, by mistake, the enzyme DNA polymerase can insert a wrong complementary base to the existing pair of complementary base pair, the nucleotide. But those type of mistake, cells came up with the many different mechanisms of self-corrections, like proofreading functions. But still, that is not perfect, those measures. So mutations will occur randomly, no matter what. And the next topic is the RNA as the first genetic material. And okay, let's quickly go through this and then finish this session. Probably what is a good end point of this lecture series. So here, let's talk about the possibility of, up until now, we all actually, the oldest highlight has been on the DNA as their genetic material. However, in the very first type of cell like a LUCA, the DNA might not have been the genetic material is the idea here. So in these modern days, RNA is being made by as a result of a copying activity of a certain area of the DNA. So this is model in RNA being copied from one area of DNA, and this is an enzyme performing such activity. But this so-called RNA world hypothesis states that it is not the DNA, but RNA was the genetic material at first, when those first cells were around in the early Earth. Why? Because if we consider DNA as the genetic material, in the first days of the cells, the obvious logical problems occur, because for each step of a DNA function requires the presence of the enzymes, and we all know that enzymes are made by proteins. So this is a diagram that we can realize the problems here. Like DNA, in order to replicate DNA, you need the DNA polymerase, a specific enzyme that is made of protein, obviously. And further, in order to make the RNA, which is basically a complementary copy of a part of the DNA, you require a specific enzyme, the RNA polymerase, which is made of a protein. And starting from RNA, by taking this information carried by RNA to produce eventual protein, obviously you do need another type of enzyme-like structure, ribosome. So in other words, the point being here is all these different enzymes are made of proteins, but to do so, you have to go through this particular procedure, which requires all this presence already, the existence of the pre-made protein, but you have never even taken a single step yet to reach to that point. So how can that be possible? So DNA shouldn't and couldn't be their first genetic material in the first type of cell, is the idea. But then why RNA? What's the special of the RNA? But instead of the DNA, RNA can function as an enzyme at the same time, is the idea. What? And what's the evidence? The existence of ribosome. Ribosome, the term ribosome is coined by this ribonucleic acid plus enzymes. So like RNA-like enzyme. So ribonucleic acid is the full name of RNA and plus enzyme, which means some RNA that can function as an enzyme is the ribosome. The existence of a ribosome first discovered by these two independently by two different renowned scientists. One is Sidney Altman in Yale University, and another scientist was Thomas Cech, but at the University of Colorado at Boulder. It sometime in mid 1980s it was for the first time discovered. So certain RNA can function as an enzyme rather than simple nucleic acid. Boring is the point here. Why RNA, whereas DNA cannot? Why RNA can function as an enzyme? It's because of their specific property. Unlike DNA, RNA can have this some specific folding procedure. We call it as a secondary and tertiary structure. What I mean by that is DNA, once they are made and they rather want to establish their complementary base pairing between two different strands. However, in the case of RNA, due to the unique property of individual nucleotide, actually the fact that it contains the oxygen in the position number 2 of the 5 carbon sugar ribose, so they prefer to have this complementary base pair internally certain within the region of the same strand, like this. We call that as a folding, RNA folding, like this. So as you can see, this complementary base pair was formed in this area, and also the partial complementary base pair was done in this particular area, and together with those areas where the complementary base pair cannot be established. So with this complex, the form of partial complementary base pairing in a region, RNA can be folded into, then it's two-dimensional. If we look at this structure from the two-dimensional aspect, then we recognize this as a secondary structure. However, everything is in 3D structure, so that it will have some unique shape like this structure. And this is one such example of like one particular kind of RNA called the transfer RNA has its own unique shape due to its folding as a result of having their internal complementary base pairing within a specific area of the same RNA molecule, and then eventually will have this specific shape. Why this specific shape matters? Because, as I said, specific shape means specific function. Yes. So any unique function is not something that's special, but only coming from their respective unique shape. And that's why usually proteins are the one that provide their specific function of an enzyme, is because protein is, due to its nature of like being assembled from 20 different, randomly assembled from the combination of the 20 different amino acids, it is much easier to generate different shapes, and some shapes are very effective in creating the environment for chemical reaction, and that's why proteins are very good ideal material for providing the enzymatic function. Is that, and if RNA can behave similarly, then why not RNA? And that's why the reason why you get to see some the RNA behave as an enzyme, and the existence of ribosome in these days. Yes, many ribosomes. So actually many RNAs functioning as ribosomes have been discovered ever since. Some examples are like this. So RNA strand can also make this complementary base pair with another RNA strand. So RNA double strand is also possible, right? So it means RNA not only can behave as a genetic material, being able to do this self-replication, but at the same time, that same RNA can behave, theoretically speaking, can function as an enzyme. So that's why scientists believe that the first type of genetic material must have been the RNA instead of DNA. However, it prefers the so-called intra-strand complement base pair due to its unique property of. And also RNA is chemically very unstable, unlike DNA. So DNA is very stable. So like the presence of this oxygen at each point will cut off this, making the strand of RNA very short-lived ones. So in other words, so with this unique ability of enzymatic functionality, at first RNA may have been the genetic material. However, once DNA became more, like once enough proteins are made, and then eventually DNA must have actually taken over as a more stable form of genetic material throughout the evolution, is what has happened. So these days actually RNA is only performing the second fiddle, the supporting role of, like, some type of RNA is acting as a messenger RNA, which is carrying the copy of some portion of the DNA, and some part of some other types of RNAs are found in the ribosome. Ribosomes are, by the way, is that big structure within which the protein synthesis is taking place. And transfer RNAs, on the other hand, are the individual small RNA that carries individual amino acid. Each different amino acid is being carried by the transfer RNA. So if you look at the site of protein synthesis, this is a big ribosome, inside of which is a ribosomal RNA is present, and its transfer RNA is carrying a specific amino acid to this site, and this messenger RNA is the one actually carrying the actual information of what type of specific amino acids should be built. And like I said, this messenger RNA, mRNA, is an exact complementary copy of the area of the gene of the DNA. So that's how it is. Although more different types of RNA do exist, but I won't go into that at this point. Simply here, only the RNA's unique property of RNA as compared with the DNA, so that with that, RNA might have been the first genetic material in the beginning. It was the only the message that need to be taken. And so with that, the small chemical evolution, we close the chapter, and then now we go into the bigger Darwinian evolution since then on, and which will be followed in the next week's lecture.